Gene therapy is a technique for correcting defective genes responsible for disease development.
The replacement of an abnormal disease-causing gene with a normal one inserted into the genome, a carrier molecule is used to transport the therapeutic genes through the body. This is called a vector. The most common gene therapy technique is when a non-functional gene is replaced with a normal gene in a non-specific location within the genome. An abnormal gene can be repaired through specific reverse genetic mutation in order to restore its original function. The particular gene can be regulated, which means it can be turned on or off.
Viruses are used within gene therapy, in which case they can be manipulated to carry the therapeutic genes in order to replace the disease-causing genes. The most common way of utilizing gene therapy is through the alteration of a virus to carry normal human DNA. Some viruses used in gene therapy include retroviruses, adenoviruses, adeno-associated viruses, and herpes simplex viruses. All but adeno-associated viruses include double-stranded DNA in their molecular structure, but are all different vectors of gene therapy. Retroviruses are used to copy the DNA of their RNA genomes. HIV, or the human immunodeficiency virus, is an example of a retrovirus. Adenovirus is a main inducer of common human colds. It is a class of double-stranded DNA that causes respiratory, intestinal, and eye infections in humans. The last of the double-stranded DNA viruses used in gene therapy is the herpes simplex, which infects a particular cell type in humans. Herpes simplex is a common human pathogen that causes cold sores. Adeno-associated viruses are a single-stranded DNA virus that can insert their genetic material at a specific site on chromosome 9.
Rheumatoid arthritis is an example of how scientists have been able to insert genes to help reduce the pain of arthritis throughout the body. RA is a classic autoimmune disease that, for unknown reasons, causes joints to become swollen and inflamed. This painful health complication causes restriction of joints and eventually can lead to immobility of them. A study in Germany has proven that when injecting beneficial genes into the cavity of the joint, it can reduce the pain and other symptoms that go along with rheumatoid arthritis. Gene therapy also allows parents to have the option of essentially helping their child's way of life in the future if their prognosis is not as healthy as a child. Is not as good as a healthy child. Diseases such as MS, ALS, cystic fibrosis, hemophilia, autism, and Down syndrome can impact a person's life greatly, along with the people around them. If doctors are able to detect the abnormal gene that causes these diseases and are able to manipulate them to modify the genetic makeup to improve the unborn child's life, why wouldn't they take the opportunity to do so?
A debate arises when the controversy behind the gene therapy exists when the question comes into play about whose disability qualifies to have such an expensive process done on them. Another issue is brought to the attention of gene therapy seekers when the cost of gene therapy comes into question, whether it will be covered under medical or if it will be miscellaneous and it would have to be paid for by the individual. The last ethical issue would involve the quality of life of the person who has the disability, and if it is demeaning to their life, and if they're trying to fit the perfect view of society and how one should be able to live their life.
DNA fingerprinting has ensured the safety of us and everyone in society for many years. This is because of the fact that every individual has their own specific print. There is around 1 in a billion chance that 2 people will have the same fingerprint. This has impacted our lives because of what this technology has done to keep our society safe from people evading the police after being charged with certain crimes. It can ensure that an individual will not be mistaken for someone else's identity based upon their physical features or any other factors, because of the fact that the fingerprints will not match up. This will ensure that there is a minimal chance of unfair trials, mistaken charges, wrongful convictions, and will lead to a just society. This impacts everyone in society on a daily basis, whether it is acknowledged or not.
Another way in which genetic engineering has affected our lives is within the food that is fed to livestock in regards to looking after their health. Livestock are fed grains, which in some circumstances farmers will put in medication, other vitamins, and minerals into the livestock's food to ensure they do not get sick and decrease their chances of getting diseases. This will ensure that animals' lives live long and healthy. However, when this animal is then at the point where it is slaughtered and then the situation flips over to human consumption, the possibility of the medicines or other chemicals within the food that was once eaten being within the muscle tissues and other edible areas of the livestock, humans will consume this as well, and all that was in the food will now be in the human system. This could have a negative impact on the health of people in general because the chemicals within the food would be toxic to humans. This is a major issue when bovine spongiform encephaloplasty (BSE), or commonly known as mad cow disease, became an epidemic problem. This was contracted by humans through eating the brain of the infected cow.
If the treatment is successful, then the individual who sought gene therapy treatment will have the ability to live a long and healthy life. They can go about having a life that does not include having to worry about something that used to impact their everyday lives and functions. This will greatly impact their life experience by increasing their ability to function as a healthy, normal member of society.
One of the negative long-term effects of gene therapy is the immune response system is designed to attack its invaders. This can make it difficult when trying to repeat gene therapy treatment because the response to gene therapy invaders will be an even faster to attack than the first time. To put simply, the risks of gene therapy are always high, but when attempted for a second time, the chances of it being successful are even smaller than the first time. Another long-term negative feedback would be the potential for the viral vector to retain the ability to cause the disease.
Yes, we agree that genetic engineering should continue because a lot of the studies are to benefit our quality of life in regards to the medicines and vaccines, preventing future generations from getting diseases that we have learned how to prevent, DNA fingerprinting giving us a safer society, gene therapy which could pave a road towards a better quality of life for people who are living with disabilities, and furthering our own scientific discoveries in the long run, while taking into account the complications that follow with each step forward.